the shameless plug for today. We will talk a little bit in that class about transcription, but only very briefly. So today I wanted to continue talking about transcription, particularly in terms of how transcription gets started. This is exactly the same thing as we started with last time. The DNA-dependent RNA polymerase, together with the specificity factor, in bacteria it's the sigma factor, binds to a promoter, and that's that specific sequence in the DNA. After it does that, goes through a couple of molecular gyrations, ends up in an elongating complex until it gets to the end of the transcription unit. It will then stop, everything falls apart, and you start again. So this is, again, for what we see in bacteria. What you see in eukaryotic cells is actually extremely similar. Also have promoters, also have a board of initiation. Just the factors that are used are a little bit different. So let's look at the bacterial promoters. And so this brings me to another concept that we really haven't talked too much about before. And that's the whole concept of a consensus sequence. So how do you get a consensus sequence? Basically, the way that you do this is you take lots and lots of promoters that you have figured out all kinds of different ways, that some of which we talked about, some of which we haven't. And then you literally line them up with each other, one on top of the other. And then just count the frequency of any particular nucleotide at any particular position. So here, also I need to give you some nomenclature. -10, -35. That's from the position that transcription starts. So, and transcription works on one strand because you're copying the template strand. And so you're going to be going in a particular direction. Again, 5' to 3', just like all polymerases. And if you then have that as your +1 position, if you go 10 nucleotides, what we call upstream of that, so -10, you end up with 75% of the time at this position you have a T, 75% of the time at this position you have an A, and slightly less than that of a T, etc. across here. This is what's called the -10 sequence. And then -35 sequence, 25 nucleotides upstream of that, you have mostly T's and G's and ACA. Now, I do not expect you to remember all of these sequences. The important thing is that this is your Frankenstein. It's the assembled, all of these different sequences. Very few of them are actually going to have the exact sequence, TPG, ACA, 15 to 20 nucleotides, TAT. But they'll have some variation of it. And again, on average, this is the most frequent of all of those. So that's the whole idea of a consensus sequence. So you have a consensus sequence, and then variations on that different theme. Another way of describing this is what we have down at the bottom. It's also called the sequence logo. And that is that at these two positions, you have lots of T's and A's, and this position you have T's, but other places you have less frequent ones. And again, the same thing is true for the -35. So why do we care about consensus sequences? Well, it turns out that the consensus sequence turns out to be a really good binding site for the DNA-dependent RNA polymerase together with its specificity factor. So if you have a sequence which is very similar to the consensus, or actually exactly the consensus sequence, you'll have a lot of binding of the RNA polymerase to that particular promoter. If you have a lot of binding of the RNA polymerase to that particular promoter, you're going to get a lot of transcription from that promoter. So a consensus promoter is usually going to be quite a strong promoter. If, on the other hand, you have a lot of differences between the consensus sequence that we have here and a promoter sequence, the RNA polymerase is not going to bind there as well. And if it doesn't bind there as well, it's not going to give you as much transcription. And so we also call this a weak promoter. So that's sort of the concept of a consensus and strong promoters and weak promoters. It really has everything to do with the DNA binding of the RNA polymerase, and in the case of bacteria, that's going to be your RNA polymerase plus sigma factor, which is the specificity factor. Turns out that this is going to be true for all kinds of different things, not just promoters, any kind of DNA binding interaction. If you have a consensus sequence, your protein, your complex, whatever it is likely to bind very well there. It's not a consensus, it's not likely to bind there. Terminators is another way of thinking about that. The other thing to mention here, this is particularly true again for bacteria, is that these sequences, -10, -35, this is where the RNA polymerase is going to bind, and it's going to determine where you get transcription. So it's not so much the starting position is +1. I like to think of the starting position as being 10 nucleotides downstream of the -10. Because where you have a promoter is completely dependent on where the RNA polymerase binds. And the fact that you've got a -10 and a -35 means you have a direction. So if you have -35 and -35 and -10, that means 10 nucleotides downstream of the -35, or 10 nucleotides downstream of the -10, is where you're going to have transcriptional start. If it were flipped around, then you'd be going in the opposite direction. And again, it's because you have a template, you're just going to be copying one of those strands. So that's what I mean by directionality. This is another way of looking at it here. Transcription is directional because it's just using one strand. So if you have a RNA polymerase on this template, completely artificial, as you can see, which is moving along here, and moving along a template strand, this is 3' to 5', is going to be making CCCC. On the other hand, if you have an RNA polymerase moving in the opposite direction, it's going to be making GGGG. So it's all dependent where that RNA polymerase is going to bind and in what orientation it is, -10 versus -35. Yeah?
Can I go either 5', 3', or 3', 5'?
Okay, so the question is, can you get, and I'm going to paraphrase here, transcription from 3' to 5' versus 5' to 3'? And the answer is no, it's always 5' to 3. So here, if you look at where this is being made, this is 5' to 3', so CCCC. And here, it's 5' to 3', GGGG. It's going on that opposite strand. Make sense? Okay, so this is just an example of this. If you just look at a pretty typical genome, this is E. coli. We have different genes that are being transcribed off of the different strands from particular promoter sequences. So here's a promoter sequence right here. And it turns out that this promoter sequence is going to give you transcription going in this direction and in this direction. And it turns out it's actually quite common that you have promoters that will give you transcription going in both different directions. But you can also have examples here. Very often these are shown as little green arrows. Here you're getting your transcription starting here, etc. But really seeing both strands. And again, it's just completely dependent on where that RNA polymerase binds and in what orientation it binds, which of those strands ends up being transcribed. Once you start transcribing, I mentioned this last time as well, the elongating RNA polymerase complex, really, because it's got not just the RNA polymerase, but a number of other proteins that are associated with it. Fortunately, people don't call this the transcriptosome. That would be even worse than most of the other zones. But it's, again, it's the nucleotides plus the proteins. Here we have a relatively low resolution structure of the DNA-dependent RNA polymerase. A couple of important things about this here. One is that there's this piece of the protein that kind of loops over one of the strands, and this is actually the non-template strand, and really serves to pull those two strands apart. So we talked about open complexes and closed complexes last time. Open complex is when the two strands are apart. Closed complex is when the two are together. In order to be able to transcribe, i.e. make an RNA copy from the DNA, those two strands have to be apart and have to be held apart. And that's the process here that's being done through this, what's also called the rudder structure in the RNA polymerases. Holding those two strands apart and moving the template strand away from the non-template strand. One thing that hopefully this will kind of look like, you kind of wrapped around one of those strands. So this looks a lot like some of the helicases that we talked about before. It's not completely around, but partly around. And that means that you've got very highly processive polymerase. So the RNA polymerase, again, as I mentioned last time, unlike DNA polymerases, first, it doesn't need a primer. And second, it is highly processive once it gets into elongation mode. Last time we talked about abortive initiation. And so abortive initiation, you spit out a few nucleotides of RNA every time when you start. But once you have this conformational change, then it's a highly processive polymerase. So this is the elongating polymerase. It will continue. Now, of course, once you've got a highly processive polymerase, you have the problem of how do you stop when you get to an end. And there are multiple different ways that this happens, but the easiest one to think about conceptually, I like to call the hairpin loop structure. This is a structure that forms in the RNA. So this is the newly made RNA. Forms these hairpin structures. And hairpins are just base pair interactions that form in the RNA together with a whole stretch of U residues in the RNA, which of course means that they're base paired to a bunch of A's here. Just like AT base pairs, UA base pairs only have 2 hydrogen bonds, so they're slightly less stable than you have with GC base pairs. So what seems to happen in this case is you have a secondary structure which forms in the RNA, and the formation of that secondary structure seems to be strong enough to pull the rest of the RNA away from these weak UA base pair interactions to pull the RNA out of the RNA polymerase structure. And once it's pulled out of the RNA polymerase structure, then the RNA polymerase will dissociate from the DNA. You start the whole process again. This seems to change the structure of the RNA polymerase. Exactly how it works is still a kind of open question. People are still trying to figure out exactly how this works. I've heard people call this the speed bump plus oil model for getting off of the particular template. Basically, the speed bump would be this secondary structure, and the oil is the UA base pairs in terms of that process. So this is basically all we're going to talk about in terms of transcriptional initiation in bacteria. There are some exceptions. We talk about regulation. We'll talk about those a little bit later on. But as far as the general mechanism, that's it for bacterial transcription. We're all happy about bacterial transcription. So happy about it that we can grab our clickers and take a look at it. Okay, so switch over here. Ridiculously wordy question. Sorry about this. If a -10 sequence in a bacterial promoter is a palindrome and there's no -35 sequence, then what would you expect? There would be no transcription. There would be transcription on one strand downstream from the -10, one strand upstream from the -10, both strands downstream from the -10, both strands upstream from the -10. Give you a minute to think about it yourselves, and then you may go beyond that.
If there was going to be transcription at all, it would be downstream. Yeah. So, but if it doesn't have both,
we don't quite have equal distribution across all answers yet, but let's just stop and have people talk about what they think the answer is.
up right now.
It's getting quieter. Let's try again.
Oh no.
Yeah, feel free to discuss while this happens, and make sure you press the button. If you don't think it's working, press it multiple times.
There is no reality which way it goes.
You got more time?
Yes, more time, please.
More time, please.
More time.
Give me another 30 seconds. What do you think the answer is?
Do you think it's A? No transcription at all?
I think that's what I put,
I'd
but
say it was a bad question because whoever was thinking about it wasn't very clear on their terminology. And
consensus, termination, directionality. If there's no, I think it's no. I think it's none. Because there's no minus 35, but I'm not sure.
Okay, we'll stop it now before showing the answers so that everybody doesn't get a chance to vote afterwards. Rookie mistake last time, even though I can't really claim to be much of a rookie. So before I show this, I'm just going to block all the rest of it. A couple of important things about this question. The first one is palindrome. So what's palindrome mean?
It's the
Same way of reading forwards or backwards. So what does this mean about directionality of transcription? It could really go one way or the other way. Because it's only dependent on where the polymerase is going to bind. So you could bind this way around, it could bind that way around. So what does that leave us with? D or E, right? Can't go for C because that would be the easy one. That's what Stedman always does. So it's going to have transcription on both strands. And what's the definition of downstream and upstream? So it's the opposite way from where your minus 10 is, or the direction that your minus 10 is going to your plus 1. So what's the correct answer?
D.
The correct answer is D, yes.
You were right the first
So
time.
only
No second guessing, Melissa. Oh my gosh, what a curve.
I was told they should be harder questions, right? Harder questions for clickers? No. Okay, so I'm going to shift gears a little bit. We'll talk about eukaryotes. Turns out that eukaryotic transcription is actually one of the more different kinds of fundamental processes that happen in molecular biology between bacteria and eukarya. And yes, we'll talk about archaea, don't worry, we'll get there. So transcription in eukaryotes, again, this is DNA-dependent RNA polymerases. There are 3 related RNA polymerases that you find in eukaryotic cells. And again, the molecular biologists are incredibly creative. They name them 1, 2, and 3. The one that we mostly are going to care about is RNA polymerase 2, because that's what makes almost all of your messenger RNAs and a bunch of these other interesting regulatory RNAs. But there are a couple of other polymerases. And probably why we have these different polymerases. It has to do with the fact that, we talked about it last time, the majority of RNA that you have present inside cells is ribosomal RNA. It's So you need a whole bunch of ribosomal RNA. So eukaryotes have evolved to have a specific polymerase that makes most of the ribosomal RNA genes. And so that's a very specific polymerase. Actually binds to different promoters. And those are the promoters that are important for most of the ribosomal RNA genes. What's the second most common RNA that you have in cells? tRNAs. So you have an RNA polymerase which is specific for tRNAs. Also different genes, and turns out also encodes some of the ribosomal RNAs as well. So RNA polymerase 1 and RNA polymerase 3, those are the ones which are making the really common RNAs that have relatively boring structures, but you need to make lots and lots and lots of them. On the other hand, RNA polymerase 2, this is the one which is making all of these different protein coding genes. And this is where you're getting all of the regulation that takes place in which genes are turned on, which genes are turned off, what genes you make large amounts of, very small amounts of. And so this is the one which is really where you get all of your regulation. So we're going to really focus mostly on the RNA polymerase 2. But all of these RNA polymerases are actually related to each other, not just RNA polymerases 1, 2, and 3. Turns out the bacterial polymerases are related to each other. We'll take a look at their structures in just a second. But even if you look at the sequences of these different subunits, bacteria have 5 subunits: beta prime, beta, alpha. Which one is this? Omega and alpha 2. These are all homologs of genes that you find in eukaryotic RNA polymerase 2 and also 1 and 3, but also you find in archaea. And one of the things about archaea that's hopefully really obvious here is you have all of these subunits of DNA-dependent RNA polymerases, which are much more similar to eukaryotic, and particularly RNA polymerase 2, than to the bacterial different genes. But overall, these guys are extremely similar to each other in structure. And so these are high-resolution structures of DNA-dependent RNA polymerases from bacteria, archaea, and eukaryotes. This one, if I remember correctly, is from Sulfolobus. This one is from Thermus. And this one is from yeast. They all have a basically similar overall structure, sort of this clamshell structure. And these are the two largest subunits of this multimeric protein, beta and beta prime. And 1 and 2 here, A and B, etc., across all of these different organisms. The archaeal and eukaryotic polymerases have these extra pieces hanging off of them, but fundamentally they're exactly the same in terms of their overall structures, which is not terribly surprising because what do? They take a DNA template and make RNA from it. So fundamentally, in terms of their activity, they're going to be extremely similar relative to each other. So let's look a little bit at the RNA polymerase 2 structure. RNA polymerase 2, one of the first differences is it doesn't have a sigma factor that's associated with it. So the question immediately is, if you don't have a sigma factor, but you still need to bind to a promoter, how do you do that? So these RNA polymerases need these things called general transcription factors. So factors, again, just your fancy molecular biology terminology for proteins. And these are all called, again, ridiculously creatively named, transcription factor associated with RNA polymerase 2, so TF2, and then a whole bunch of alphabetical things, A, B, D, E, F, H, etc. So why does eukaryotic RNA polymerase 2 need all of these extra factors? Well, one of them is clearly specificity, and that turns out to be the TF2D, particularly the Tata binding protein that we'll talk about more in just a second here. But also has to deal with nucleosomes and the fact that in general, eukaryotic genomes are way, way, way bigger than bacterial genomes. There's a lot of junk DNA. And so how does the RNA polymerase deal with a lot of this junk? And so probably the specificity of a lot of these general transcription factors is very important. So we'll look at promoters themselves in just a little more detail in a second. The main part of promoters is what's called this Tata box. Why Tata box? Because it's got lots of thymidines, adenines, adenines. Again, we'll look at the sequence here in just a second. And this Tata box is bound to the Tata binding protein. Again, ridiculously creative, but at least tells you exactly what's going on. The Tata binding protein is part of TF2D, transcription factor 2D. And may or may not make this easier to remember. The reason they're called A, B, C, D, E, F, etc. is that's just the order that they come off of a chromatography column. So fraction A, fraction B, fraction C, etc. So TF2D has this DNA binding subunit. It's actually a separate protein by itself. Again, associated with TF2D. There are lots of other proteins associated with TF2D that we'll take a look at in just a second. The second protein that also binds specifically to DNA is TF2B. So TF2B binds to the TF2B recognition element. So elements we haven't really talked about too much yet. Element is just a DNA sequence. So a element is going to be DNA sequence. The factor is going to be what interacts with it. So the TF2B recognition element is a binding site for TF2B. You also have the main one that we'll talk about here, TF2H. The way I remember TF2H is TF2H has a helicase activity. And it turns out that that helicase activity is absolutely critical for transcriptional initiation, pulling those two strands apart. The helicase, we talked about the helicase activity before, going from closed complex to open complex. In bacteria, that's actually part of the polymerase itself. In eukaryotic polymerases, they need this extra protein, TF2H. TF2H also has a very strong kinase activity. That kinase activity phosphorylates the RNA polymerase on something called the CTD, the C-terminal domain. So it's a separate structure, again, in your RNA polymerase. It's actually part of the largest subunit of the RNA polymerase. Has lots of sites that can be phosphorylated. And part of that phosphorylation seems to be important for getting this complex that assembles here to move away from the promoter and start transcription per se. So this is the cartoon version, got the table version right here. TF2D, really care about this Tata box binding protein. So TBP is one of the subunits of TF2D. There are lots of other subunits that are associated with TF2D. These are important for not just recognizing the Tata box. Other parts, as we'll see, turns out that TBP is sort of the last part of TF2D that associates with the DNA. There are other parts of TF2D that interact with other parts of the promoter, and also many other interactions with other proteins, transcriptional regulatory proteins, interact with these so-called TAFs. So what's a TAF? It's a TBP-associated factor. Yes, it's Russian doll kind of thing. So, but these are part of TF2D. TF2B, straightforward, only one subunit, binds to the TF2B recognition element. The TF2B recognition element, we'll see in just a second, this is really important for getting the orientation of transcription. So again, like you have a palindromic -10, turns out that Tata boxes are almost always palindromic. So if you have a palindromic sequence, your RNA polymerase doesn't know which way to go. The B recognition element usually gives you that particular stability. We're not going to talk about the roles of TF2F or TF2E, even though one of my really good friends in graduate school, spent most of her PhD working on actually cloning this particular gene. TF2H, again, has this helicase activity and phosphorylation activity. So what do these promoters look like? This is a consensus promoter for a eukaryotic system. One of the things you'll notice here, we don't have really nice long sequences like you have in bacteria, -10 and -35. We just have relatively short sequences, and they're actually not very well conserved at all. So look down here, these are our consensus sequences. Tata box, TA, and this slash means about half the time an A and half the time a T. So that's actually the one which is best conserved, is the Tata box. But then this TF2B recognition element, sometimes it's G, sometimes it's C, sometimes G, sometimes C, sometimes G, sometimes it's A. And then we've got these 4 bases. 4 bases are not very much if you think about it in the context of a 3 billion pair human genome. So not a huge amount of consensus sequences. There's what's called the initiation site, also very poorly conserved, and you can see this N in the middle here. N means any 4 of the nucleotides. And then there's a downstream promoter element, again, also not terribly well conserved. One of the things that might be a little surprising here is we talked about the TDP, right? That binds to the Tata binding, the Tata box, which is about -30 nucleotides from the transcription start point. But then TF2D is interacting with this INR sequence, 30 nucleotides away, and this downstream promoter element, another 30 nucleotides away, 60 nucleotides away. That's a really long distance for one factor to actually be interacting with. And it turns out that literally just last year, people finally got a good idea on what was going on with this. But before I want to start with that, let's talk about the Tata box and the Tata box interaction. And the first example I wanted to use here was, again, kind of a giveaway when I was in graduate school. I actually still have the original of this particular article. This was a big deal in November of 1992. Anytime you have a picture on the cover of Nature, it's a big deal. So this was the crystal structure of the Tata binding protein, Tata box binding protein. And look, it's got this wonderful double helix right down the middle. Clearly that's the way it's interacting with DNA. This is all wonderful and good, except for the fact that it was wrong. A year later, they published the crystal structure of the DNA and the Tata binding protein. And hopefully you'll notice a slight difference between these two. DNA straight down the middle, or very bent, and with the Tata binding protein really kind of sitting as a saddle on the outside of it. Another thing here, which is rather important, look at the DNA. Is it nice and straight? No, it's very bent. And this bending will mean that the pieces of DNA that used to be a long way apart, remember, -30 going up to +30, these pieces are now going to be a lot closer to each other because of the binding. It turns out the process of binding of the Tata binding protein really does very heavily bend that piece of DNA. So this is not shown in our textbook, but I wanted to show the original articles here as well, partly because everything that you see on the cover of Nature might not be right. So this is, again, where that particular paper came from, and this is now a more of a cartoon version. Tata binding protein is a dimer, has these alpha helices and beta sheets, and the binding of DNA by this Tata binding protein, and you can see TA right here. ATs are easier to bend, and B, a lot of the interactions here are actually in a minor groove of DNA, and it's this folding of the DNA into a particular structure that seems to allow all of the other proteins to actually associate with it. So that's just looking at TDP. What about the rest of TF2D? And so that's when I wanted to click through and take a look at this, because this is from a group at Berkeley from, again, I think last year. I'll go to this link. and sometimes it works, sometimes it doesn't. Okay, I'm not going to go to that link. Okay, I'm going to have to copy and paste this. Hang on a second. See my family here briefly. Start. For some reason, I want to I actually sent them an email and said, "Oh, you have an old version. You need to buy the new one." Thanks. So this is, should be anyway. Back up, reload. So this is a paper from literally last year. We'll skim ahead a little bit here. But so here's you have the transcription. This is your RNA polymerase, high resolution structure thereof, making your messenger RNA, etc. Making lots of copies of it all throughout the different parts of the genome. Again, they need to bind to very specific promoter sequences in order to make sure that you have the appropriate transcription from the appropriate place. And it's this association with the promoter that's what's going to determine where you get transcription. And so this is a much more higher resolution part of that whole structure. This gray part is the RNA polymerase. This one, remember correctly, is TF2H. And then TF2D. And particularly it's this TF2D structure. Notice multiple different polypeptides that are associated with it. That's the Tata binding protein. And one of the things you may notice here is this guy's moving around a whole lot. So starts out over here and then ends up all the way over here. These are the Tata binding protein associated factors. All the rest of TF2D is made up of these things. So the first thing that seems to happen is you have interactions, again, down here. Downstream promoter element, initiator element. And then, and only quite a lot later, is when you have the TBP that associates with the Tata box and then bends the DNA. And so what this particular group, Eva Nogales' group at Berkeley has been doing is looking at the structure. And so this is the highest resolution structure so far of the TF2D complex. And again, the Tata binding protein is sitting up here. This is going to be moving quite a bit in terms of then interacting with the promoter later on. So where are these? They've got all kinds of different, TAF6, et cetera, et cetera. These details are not really critical. Just wanted to show you how this really interesting conformational change is happening when you get the interactions with the DNA and with the promoter. So TF2D, TAF4, TAF6, TAF9, we now know a lot of these particular proteins, but again, the TBP, right up here, this little red segment, which ends up all the way over here, binding to the Tata box, and you see making a really big kink in the DNA. But the way that this starts out is it actually will interact much further downstream with the Tata box being way over here, downstream promoter element. This guy moves around, and eventually you get association of the TBP together with the promoter. So TF2D is really where it's at in terms of getting everything together to work properly. Okay, so it's worth going through that. In fact, Eva Nogales gives a little bit of a background commentary on how all of this works, probably better than I do. You can go and take a look at that on YouTube. So that's TF2D, but we've got TF2D, we've got the RNA polymerase, we've got TF2H, we've got TF2E, we've got TF2E, we've got TF2F, and as if that weren't enough, there's actually a whole set of other proteins, particularly this set of proteins called the Mediator Complex here in purple. And this Mediator Complex in purple, this is what interacts with activator proteins. And as we'll see when we talk about transcriptional regulation, activation is really key for getting eukaryotic transcription to work. You get all of these proteins assembled at a promoter, and actually usually you don't get any transcription that happens until you get activation. So people are still working at trying to figure out all of the processes that are happening here. You also have chromatin remodeling complexes, histone acetylases. This is a ridiculously large complex of proteins that is going to be generating your messenger RNAs in order to then make proteins, etc. You may or may not remember when we talked about chromatin structure, where you have active polymerization going on, active transcription, it's decondensed, or it's moved to a different part of the nucleus. Well, it's not surprising that if you have to have these ridiculously large complexes, it's probably actually easier to move the DNA around rather than moving all of this crazy structure around. So it's the DNA which is moving and moving through these, people also talk about them as transcription factories rather than the transcription factory binding to the DNA and then cruising along it. However, once it cruises along it, you still have the same kind of problems that we talked about when we talked about replication. This is eukaryotic DNA. Well, eukaryotic DNA is in chromatin. It's bound to nucleosomes. We'll talk a little bit about nucleosomes later on. Turns out that nucleosomes are okay, but even more of a problem is the fact that you need to pull apart the two strands. Remember, when you're unwinding DNA, that also causes supercoiling effects. And so if you don't have free ends, then you can end up with lots of problems with supercoiling. And so this is just another way of looking at that. Here we have something pulling apart the two strands. If you're moving in one direction and each of these ends are fixed, which we know is happening because of the nuclear scaffold and the loops which are present in genomes, that you end up with these supercoils that happen. So you actually do need topoisomerases also for transcription. One advantage of this positive supercoiling, so if you're pulling things apart, you're going to be making, going from wrapped to unwrapped. Wrapped to unwrapped actually generates a positive supercoil in front of where your protein is moving. Turns out that positive supercoiling is actually really good for removing nucleosomes from DNA. So positive supercoiling helps get that transcription, but you also are going to end up with all of these supercoils that will eventually stop the movement of the polymerase. So there are some nice experiments where people look literally at polymerases moving along DNA, and if the ends can't move and there's no topoisomerase, that polymerase will get stuck. So you do absolutely have to have these topoisomerases in order to get transcriptional elongation. I've talked too much. Time for you to talk amongst yourself, right? No.
I thought DNA gyrase was the same thing as topoisomerase.
Okay, so the question is DNA gyrase. So I just threw up DNA gyrase here. DNA gyrase is actually a topo 2, type 2, and is present in bacteria. It deals with negative supercoils and actually takes out negative supercoils. I just wanted to throw it up here, and thank you for reminding me, is that it's not a problem that you just have in eukaryotic transcription. Also, bacterial transcription is the same problem. You're pulling the strands apart, and you end up with supercoils. And so dealing with those supercoils, you're going to have an enzyme to do that, and particularly DNA gyrase. That's the main one which is used in bacteria. More questions? I need a break, so it's time for quick questions, which should be more straightforward, I hope, than the last one. Which of the following bind to specific DNA sequences? TF2B, TF2D, TF2H, A and B, A, B, and C.
TF2B and D. TF2B and TF2D. Guess Tata binding protein isn't an option on there, so D, which is A and B. It's too early to pull things like that.
Five, 3, 2, 1. Nah, we're not at 80%. So, talk to your neighbors. Tell them what you voted for. Why? Get to 100%.
TF2H unwinds DNA at this transcription start point. Phosphorylates. Binds to a specific DNA sequence.
Bind to specific TF2D regulates DNA binding by TBP.
Tata binding protein, which is, by definition, is recognized as the Tata box, which is a specific sequence. Well, it's close to a specific sequence. I don't know.
Ready for another round?
So,
okay, let's go.
They had D also.
They said D?
Yeah.
Okay. It's either B or D.
Yeah. But TF2B also recognizes VRE elements in promoters, accurately positions RNA polymerase at the start site of transcription, so it has to recognize that start site
On the DNA.
But maybe the start site isn't a specific. Do you think that next week you would let me bring your dog in?
I don't know. Is your dog quiet?
Yeah,
He sleeps.
he's much quieter than children.
We're not quite at 100%.
Oh,
I guess the
yay.
big question is, does TF2H bind to DNA specifically? And the answer is no, it does not bind specifically to DNA. It's brought there by the rest of the complex. TF2B binds, of course, with the recognition element, and TF2D has the Tata binding protein that's associated with it. And just to confuse you even more, I love structures, as you probably have guessed a little bit by now. That if you now look at what we think we know about it's also called the pre-initiation complex, and so this is all of the proteins that bind to a particular promoter before you actually start to get transcription. This latest version, actually it's from a couple of years ago now, looks at the structure of the mediator complex together with all of these other general transcription factors. And actually this one's missing a whole bunch of them. The main thing it's supposed to show is this mediator complex, which just, again, adds to this whole glob, and I don't really see it as anything more than a glob, of all of the individual proteins that you have to have at a transcriptional initiation complex. So that brings us to why we should all study archaea, which has to do with transcriptional initiation in archaea. This is particularly the work which is done now in Dr. Michael Bartlett's lab here at Portland State, and some of the work that I did when I was starting my postdoc. A couple of big differences between archaea and eukaryotes, but also with bacteria. Turns out that the proteins that are involved in archaeal transcriptional regulation are way more similar to what you see in eukaryotic regulation and also in binding to promoters. So we'll see in just a second, there's only one RNA polymerase in archaeal systems. There's a Tata binding protein, but there's no TAPs. So it's just the Tata binding protein. There's TFB, and this is now just TFB, it's not TF2B because there's just one polymerase. So you don't have to call it TF2B. So we've got RNA polymerase, TFB, and TBP. This is all that you need to get transcriptional initiation. So you don't need all of those extra bits and pieces, bells and whistles, but the RNA polymerase itself and the fundamentals of TFB and TBP are actually extremely similar to each other. And so another way of looking at that is this one. It's a couple of years old now, but if you look at a pre-initiation complex in archaea, the RNA polymerase is extremely similar between archaea and eukaryotic systems. Tata binding protein itself is very, very similar, but it doesn't have all the rest of TF2D, which is yellow in here. TF2B is also extremely similar. Turns out that you do have a TF2E homolog that you have in archaea, but at least in vitro, which is a purified system, you just need the RNA polymerase and these two other proteins. So way easier to study the basics of thinking about transcriptional initiation because you don't have all of these extra factors here. And just to try and prove it, as it were, to you, what you have with these, also like to call them the core transcription units being very similar to each other, the structure of the Tata binding protein and TFB from Pyrococcus furiosus, which is a classic archaea, and in fact the same archaea that Dr. Bartlett works with here at PSU, is practically identical in structure to the Tata binding protein and TF2B structures that you see in eukaryotic cells. And again, to show how old I am, Otis Littlefield, who was the first author on this paper, was also one of my buddies from graduate school. So as an overview of this process and why it's such a wonderful system to use, a much simpler version of the eukaryotic call 2 system, promoters are very similar. They've got Tata boxes and TFB or TF2B recognition elements. So the promoters in the cells are very similar. The RNA polymerases are very similar to each other. The actual number of subunits, the sequences of those subunits, the overall structure, which I showed you before, is extremely similar between the two. Big difference is in eukaryotes, you have this C-terminal domain that's on the largest subunit of RNA polymerase. You don't have that in archaeal systems, but you also don't have TF2H in the archaeal systems, which would be phosphorylating that CTD. You also don't have all of the mRNA modification systems that you normally have in eukaryotic systems in archaea either. So we'll see that. Initiation factors, what do you need? TBP, TF2B, and a whole bunch of other ones in RNA polymerase 2, whereas in archaea, you just need TBP and TFB. What about chromatin? Well, we already talked about chromatin as being an issue for eukaryotic systems. Briefly, when we talked about nucleosomes, there actually are some nucleosomes in Euryarchaea, but not that many, and certainly nowhere near the same amount of compaction that happens. So, if you want to study transcription in archaea. At least that's the hopefully take-home message here. So I wanted to finish up by just looking at this transcription movie, which hopefully we can get to run. It's still not running here. This technology, so much fun. This also has a commentary, which is really not bad either. So let's see if we can get this to actually run. Here we are. So here we have a promoter sequence, a little hard to see here with this arrow, where we have some general transcription factors bound here. And this is actually a relatively old version, i.e. doesn't have November of last year's update in it. This is supposed to represent TF2D, binding through the Tata binding protein to this piece of DNA and giving you a pretty big kink. Here comes the rest of the RNA polymerase, associates with the promoter. Now we have this mediator complex that binds on the outside. And now this pre-initiation complex just kind of sits here and waits. And waits for that activation, which is going to happen from a separate binding protein. Here it is, coming in from the outside. That's our activator protein that interacts with the mediator complex. Should do in just a second here. I think it will. Maybe it won't. Boom. There we go. And all of a sudden, we're off. So here's our DNA-dependent RNA polymerase making transcripts. That's that yellow piece which is hanging off the back. You can see that it's really stuffed itself through the 2 strands of DNA and making that RNA as it's zooming along the DNA structure. Again, if we think about this from that whole complex of where we've got all of those extra enzymes associated with it, all the extra proteins, all the extra factors, it's much more likely that the DNA is moving through this complex as opposed to the polymerase itself cruising along the DNA. Hits a terminator, that RNA is released. So I think that instead of starting to talk about messenger RNA processing, we'll just stop.